The Great Circle, Part 2. Previously, we learned that photosynthesis takes place in the chloroplast. In the light reactions, solar energy is captured by photosystems on the thylakoid membrane and used to attach phosphate groups to energy-carrying molecules like ATP. Now we're going to look at this process in greater detail. Now, something that often confuses students is that the first step in this process is photosystem 2. Don't let this confuse you, but the first step in light reactions is only called photosystem 2 because it was discovered later. Just remember, photosystem 2 is first. A photon, a particle of light, will be captured by one of the pigment molecules in the photosystem. This light energy will move toward P680, the special pair of chlorophyll molecules in the reaction center. The energy is used to split water molecules, which we call photolysis. Here we see one water being split into 2 H+ and 1/2 O2. As this occurs, the electrons that were being shared by the hydrogen and oxygen are pulled off. Now, in reality, you have to talk about things in whole numbers, you actually have to have 2 waters to get 4 H+ and 1 O2. But the basic idea is that the energy of light is used to split the water molecule. The 2 electrons, which are removed in each of those reactions, and the energy they carry, will then move into the thylakoid membrane. But at the same time, the products of photolysis, which is protons and O2, will tend to increase within the thylakoid space, which is called the lumen. The concentration of these 2 products will be greater within the membrane than without, so both will tend to escape. But H+ and O2 will leave in different ways. Oxygen will tend to leave by simple diffusion through the membrane. It'll just leak out randomly because it's small enough to slip through cracks in the membrane. But it's a random process when it leaves. You don't know when or where it's going to leave. Protons, however, will accumulate much faster. A special protein channel will help transport them across and, as you will see, do valuable work in the process. See how the hydrogens, they're leaving constantly, and they're not leaving randomly. They're all leaving out through a particular location. That's through a protein channel, ATP synthase. So the question we should ask, though, is why do the protons accumulate much faster? The electrons that are produced by photolysis will move through a series of membrane proteins, which we call an electron transport chain. These are moving negative charges. This motion of negatively charged particles helps attract H+ outside the membrane. H+ is a positive charge. And that causes those protons from outside the membrane to move into the thylakoid. Thus, the increase in protons is due to 2 factors. Photolysis, the breakdown of water inside the thylakoid, which in this illustration is shaded blue. That's occurring in the lumen. And the other is the motion of electrons within the thylakoid membrane attracts free protons outside the thylakoid. So again, as the electrons move through the membrane, they are moving negative charges, and they will tend to attract any positively charged particles, protons, from outside the thylakoid. So there are 2 reasons why the protons are attracted. And we end up with a high concentration of protons inside the thylakoid and that can be used to do work. The way this works is similar to an electrical power plant. Here we have a cross-section of such a plant. Water will flow automatically from a region of high concentration behind the dam to one of low concentration below the dam. Gravity will pull the water downhill. The moving water will turn the turbine of an electrical generator. The kinetic energy of the water is converted into electrical energy. In the same way, protons, H+, they're in much higher concentration inside the thylakoid membrane than on the outside. High concentration within, low concentration without. So as a result, protons will flow automatically out of the thylakoid, again going from high to low concentration. These moving protons will turn the turbine of a membrane protein. We actually have a miniature machine. This protein, called ATP synthase, is an enzyme that attaches a phosphate group to a molecule of ADP to form ATP. The energy of the motion of the protons of H+ has been converted into chemical energy stored in a phosphate bond. If we look at the inset here, we don't see the blades moving, but the motion of the protons, shown as positive charges, little plus signs at one side in high concentration, the protons automatically move through the ATP synthase channel. They move down. As they move down, they turn the blades, the rotor of the protein, and one part of the braid has a shape that matches ADP, one has a shape that matches phosphate, and puts them together. So ATP synthase is an example of an enzyme, a protein made by a living thing that has a complex shape, one that can make a chemical reaction more likely to happen. And that's our definition of an enzyme. An enzyme has a complex shape called the active site, which makes a chemical reaction more likely to happen. The active site fits the substrate like a lock fits the key, brings the items close together, and causes the chemical reaction to occur. Now, in the case of ATP synthase, it is a miniature motor. And this miniature motor actually rotates within cell membranes. As it moves, it attaches phosphate groups to form ATP. Here in the animation, you can see pink things coming out and blue things going in. The blue things represent ADP. The pink things coming out represent ATP. ATP synthase is so basic, so essential for the function of the cell, that it is used by all living things. All living things use this particular enzyme in the membranes of their cells to help manufacture ATP, to make a chemical reaction that on its own might never happen in a million years. But now it increases the rate of reaction and makes it so that it happens constantly. The magic of enzymes is that they lower the activation energy and they make a reaction that on its own would not happen, would not be likely to happen, and it makes it certain to happen. Now, previously, we learned that ATP is made of molecular building blocks that are already present in large numbers in the cell. Adenine, ribose, phosphate groups. Because these building blocks are already present in large numbers in the cell, they're just constantly there, floating in the fluid-filled interior of the cell, what we often call the cytoplasm. The building blocks are adenine, a nitrogenous base, shown in orange, a 5-carbon sugar, ribose, shown in green, and then a series of phosphate groups. Adenosine monophosphate by itself is the same as one of the nucleotides used in DNA or RNA. We attach additional phosphates to make either ADP or ATP. Again, this happens randomly. But to make it more likely to happen, we have the enzyme ATP synthase. And it will cause this reaction to happen. A phosphate group is attached to ATP. And it's easy for the cell to do this. It's easy for the cell to add a phosphate group to ADP and turn it into ATP, because those building blocks are always present. Again, the cell can easily add a phosphate group to existing molecules such as ATP. Adding a phosphate group is called phosphorylation. Here we can clearly see that adenosine diphosphate has two phosphate groups. Well, let's add another phosphate group to it. Now we'll get ATP. And when we compare the illustration, we can now see that there are three phosphate groups instead of two. So this is very easy to do. This is a form of energy storage because it takes energy to form the chemical bond between ADP and the phosphate group. Let's go ahead and put the two together right now. Dehydration synthesis. We're going to form a chemical bond. How do we do it? We pull out of water. Oh, there it goes. So let's look at that again. Again, we have ADP. We bring here another phosphate group. We pull off the atoms, two hydrogens and an oxygen, which come together to form water. And what's left behind is the chemical bond. And the chemical bond is formed by dehydration synthesis, as previously taught. One more time. The phosphate group coming in, you can see it has two hydroxides. They'll come together. Two hydrogens and oxygen will be pulled out as a water. The chemical bond is formed. Dehydration synthesis. Now, in the same way, if the energy is required, then instead of making a phosphate bond, we can break it. How do we break it? We put a water in, hydrolysis, and look what comes out. Not only does the phosphate group come out, but we release some energy. ATP becomes ADP, and energy that was stored in the bond is released. Hydrolysis, add a water, break a bond. One more time, let's add the water. We break the bond between two phosphate groups. It's hydrolysis. The phosphate group comes out, but also we get a release of energy. To complete the light reactions, the electrons moving through the thylakoid membrane will be boosted to a higher energy level at photosystem 1. To understand, these electrons already have a certain amount of energy, which they got at photosystem 2. But now at photosystem 1, they get additional boost. Photosystem 2 was P680. Now they are going to be boosted to P700. So these will be high energy electrons. Electrons moving at a higher energy level. These high energy electrons will then attract a proton which has enough energy to reduce a compound, NADP+, to NADPH. When we say reduced, we mean that its overall charge has been reduced. Like ATP, NADPH is useful for short energy storage. Compounds like ATP and NADPH are not stable, and they can only be used to store little energy for a few reactions at most. Long-term storage requires stable high-energy compounds. For that, a series of reactions must take place called the Calvin cycle. The Calvin cycle is used to take the tiny amounts of energy in things like ATP and NADPH, and it's going to be used to assemble sugars. It uses ATP and NADPH in the light-dependent reactions to produce high-energy molecules. This will take place outside the thylakoid in the stroma, the fluid-filled interior of the thylakoid space. The first step of the Calvin cycle is that 6 carbon dioxide molecules are combined with 6 5-carbon molecules. 6 times 5 is 30. Then this will be used to produce 12 3 carbon molecules. 3 times 12 is 36. So 30 plus 6 gives us 36, rearranged into 12 3-carbon molecules. Energy from ATP and high-energy electrons from NADPH are used to convert the 12 3-carbon molecules into higher energy forms. It says 12 3 carbons at the top and the bottom, but the ones at the bottom have more energy that's been donated by ATP and NADPH. Now, 2 of the 3-carbon molecules will be removed from the cycle to produce things. Different cells will produce different things. But whether it's sugars, lipids, or amino acids, or other molecules, the point is that you get a profit. The 10 remaining 3-carbon molecules are converted back into 6 5-carbon molecules, which are used in the next cycle. So again, if we look at the entire cycle, we put in 6 carbons from carbon dioxide to make 12 3-carbon molecules. We use the energy of ATP and ADPH to take the higher energy. We take 2 of those 3 carbons out, and the remaining 10 3-carbons, we add 1 more ATP once again, and we complete the cycle. And this all occurs in the stroma, which is the fluid-filled region inside the thylakoid. Now, this illustration summarizes the light-dependent and light-independent reactions of photosynthesis. You can clearly see that water comes into the thylakoid and light energy hits it, and the light-dependent reactions, and we have photolysis, oxygen comes out. And you can see that the light-independent reactions happen outside the thylakoid. The carbon dioxide comes in, and the products are sugars. And in between the light-dependent and light-independent reactions, we have ATP and NADPH both being cycled back and forth. So, to summarize photosynthesis, previously we learned that photosynthesis takes place in the chloroplast, and in the light reactions, solar energy is captured by photosystems of the thylakoid membrane and used to attach phosphate groups to energy-carrying molecules, like ATP. In the Calvin cycle, the ATP and NADPH, which are made in the light reactions, are used to power chemical reactions in the stroma to build molecules like glucose. Now, this completes the equation for photosynthesis, but the thing about equations is that they require equal numbers of atoms on both sides. So it might be helpful to make sure that we understand the formula for glucose. So we have how many carbons? Let's count them. 1, 2, 3, 4, 5, 6 carbons in glucose, C6. How many hydrogens? Well, there are more. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 hydrogens. C6, but H12. How many oxygens? Well, this is easier. 1, 2, 3, 4, 5, 6. So the formula for glucose should be C6H12O6. There should be 6 carbons and 12 hydrogens on both sides of the equation. And here we go. 6 carbon dioxides plus 6 waters, that's 12 hydrogens, gives us 1 glucose sugar, C6H12O6, and 6 diatomic oxygens. So now we have energy stored in the form of sugars. Well, this is very nice, but what if plants, or for that matter, people, want to release that energy? They must use a different path called cellular respiration. Our first step is we reverse the equation. The sugar and the oxygen that were the product of the original equation, they're now the reactants, and the carbon dioxide and the water's on the other side. That's our first step. Now we'll need another organelle to do these reactions in, not chloroplast this time, but instead another organelle with its own DNA, the mitochondria. So the mitochondria, just like the chloroplast, has an inner and an outer membrane. The mitochondria, just like a chloroplast, has an area that's heavily folded, the cristae, and it has a fluid-filled interior, the matrix. So the complex folded inner membranes, called cristae, are places where certain reactions happen, and they are heavily folded to increase the surface area for those reactions to happen. Within, there's a fluid-filled interior. Here it's called the matrix. Mitochondria act like combustion chambers in an engine, a safe place to burn fuel with oxygen. In a standard gasoline engine, the combustion chamber, we react gasoline with oxygen. We react it in the tiny spaces that exist where the pistons go up and down the cylinders inside the internal combustion engine. It's an overlapping area with high surface area. And here's the mitochondria, an actual electron micrograph showing the structure of the cristae membranes overlapping. It's a very similar structure. But before combustion can occur, we have to get some fuel. Here, the hexagon shapes, you should remember, represent 6 carbon sugars like glucose. Instead of reacting gasoline with oxygen, we're going to react sugars like glucose. But to do that first, we're going to have to break the glucose down. We're going to need to do a series of reactions called glycolysis first. Glycolysis is the breakdown of glucose or other sugars. Glycolysis requires an activation energy to get started, just like a candle needs a spark to get it to burn. And it will occur not in the mitochondria. It will occur in the cytoplasm, the fluid-filled interior of the cell, which is outside the mitochondria. So the first thing that happens is that chains of sugars, polymers of glucose like starch, will break them down into individual sugars through hydrolysis. Okay? We'll just add water, just like we discussed before. Now you'll have single sugars by themselves, and of course they contain stored energy in their chemical bonds, but the sugars, even by themselves, monosaccharides, they're still too big to pass through the mitochondrial membrane. So we're going to have to break it down in a series of steps that do not involve oxygen. So we add ATP to get started. That's the activation energy. A phosphate group is added. We have glucose 6-phosphate. It gets rearranged to fructose 6-phosphate. We add another phosphate. Now it's fructose 1,6 biphosphate. It gets split in half by 3-phosphoglyceraldehyde. And now we do reactions to both sides. Here comes the NAD+, bringing another phosphate group. And now we have 1,3 biphosphoglycerate. We add one more. It's 3-phosphoglycerate. We move the phosphate group to a different place. It's 2-phosphoglycerate. We end up with this intermediate phosphoenolpyruvate. And at the end, we have 2 molecules called pyruvate. So you can see it's very complicated, and there are many steps, and there's an enzyme for each step. But the important thing is it gets broken down. There will be a net gain of 2 ATP. The final products of glycolysis are 2 3-carbon molecules of pyruvate. And you can see its formula, C3H3O3. It's much smaller than the sugar. It's small enough to be easily transported through the mitochondrial membrane, where a new series of chemical reactions take place called the Krebs cycle. Just like in photosynthesis, we're going to do one set of reactions in a fluid-filled area, which is called the matrix. Here we have a cross-section of the mitochondria. You should see that the arrow is pointing to the interior, and that is the fluid-filled interior of the matrix. The Krebs cycle will begin by converting each of the 3-carbon pyruvates into a special complex called acetyl-CoA. Pyruvate will enter the matrix. Instead of adding carbon dioxide, as we do in the Calvin cycle, we take it out. The carbon dioxide is a waste product. We add a little enzyme. We create a temporary complex called acetyl-CoA. Acetyl-CoA will begin the Krebs cycle. Now, again, you can see it's very complicated. Acetyl-CoA is formed by pyruvate and CO2 being removed and all that. And as the cycle proceeds, we'll take more carbon dioxides out. Citric acid will be converted into isocitrate. We'll remove a carbon dioxide, converting it into ketoglutarate. We'll remove a second carbon dioxide by reducing it with NAD+, and that will produce a temporary complex called succinyl-CoA. So on this side of the cycle, carbon dioxide are removed. Now at this point, there's a net profit. There's a net gain in ATP. And this net gain in ATP will be used to charge an electron transport chain, just like in photosynthesis. So succinate, we remove an electron, and that makes it fumarate. We rearrange to malate, and then we remove another electron, and we'll have oxaloacetate. And that's the same thing that we added 5 carbon dioxide too at the beginning of the cycle to make acetyl-CoA. So this cycle takes place there. Now, from there, we use the energy of ATP to power up that electron transport chain, which takes place in the folded membranes, the cristae. Here we see charges being split. And what will happen is, as you can see, the electrons move through the membrane. And the big idea is you end up with positive charges on the outside of the membrane because the proton's being pulled off, and you end up with negative charges moving through the membrane because of the electrons being stripped off the reactions. And so the moving negative charges inside the membrane, just like before, they'll draw in protons. They'll create a high concentration, which, just like in photosynthesis, can be used to drive a proton pump. Instead of the electrons moving through the membrane of the thylakoid, they're moving through the membranes of the cristae, but the result is the same. We end up with a whole bunch of protons heavily concentrated on one side of the membrane and not so much on the other. And that difference in concentration, just like a hydroelectric power plant, can be used to power a proton pump. And just like in photosynthesis, that difference in concentration, the proton pump, can be used to power an enzyme, ATP synthase. Once again, we see the pluses representing lots of positively charged protons on one side of the membrane in high concentration, and they automatically flow from high to low. As they go through ATP synthase, they cause the blades to turn, and one blade has a shape that matches ADP, and the other has a shape that matches a free phosphate group, and they bring them together. And so, just like in photosynthesis, the enzyme ATP synthase, as it turns, it makes ATP on the other side of the protein. It's an enzyme. It makes that chemical reaction go faster, just like in photosynthesis. Now here we have a proton pump, and we have it next to an ATP synthase. And so you can see that we are having a situation where there's a lot of H plus on one side of the membrane, and they will naturally be transported by the proton pump to the either side. This is just like the photolysis of water. We'll end up with a very high concentration of H plus outside the membrane because the proton pump transports them across. And this difference in concentration is going to cause them to move. They can't move back in any other way because they're positively charged. And the moving H plus will cause ADP to gain a phosphate group. You're not seeing the blades, but the basic idea is that the motion of H plus brings them together, attaches a phosphate group to ADP, and forms ATP. This is how the hydrogens get back inside because they are, positively charged and can't get through the membrane out of the way. So we typically have one little enzyme, the proton pump, that makes the high concentration of H plus. The other side we have our ATP synthase. So now let's do the math. In glycolysis, in the cytoplasm without oxygen, we only get 4 ATP. In the Krebs cycle, in the matrix, without oxygen, we only get 2. But with the electron transport, look how much bang we get for our buck, 32 ATP. It's a total of 38. Since we had to spend a little ATP to get things started in glycolysis, the net yield is 36 net ATP for each glucose sugar. And you can see from this diagram that the profit involved in using oxygen is very, very high. And that is the advantage of using oxygen. You get a lot more bang for your buck.